David Hausler is a professor of biomolecular engineering at the University of California, Santa Cruz. He specializes in understanding the DNA of extinct creatures, but you won't catch him with a shovel in his hands.
Paleontologists study evolution by digging up fossils and working out how the animals changed their morphology, their shapes through the ages. We take the DNA of living animals and work backwards to what the DNA must have looked like 10, 40, 100 million years ago. We actually look at the DNA of living species, and we use the computer to take that DNA and trace it back to find out how it looked in the past and how it changed over millions of years to create the different species that we see today.
It may be a letdown for Jurassic Park enthusiasts, but real dinosaur DNA is simply too fragile to survive the 200 million years since the Jurassic period. If you want to find the DNA of extinct beasts, Hausler's revolutionary new process called computational genomics may be your only bet.
So we don't actually find the animals themselves, or their remnants, the bones, but we can reconstruct with the computer analysis what their genomes look like. And a genome is what determines the animal's shape and size and behavior.
One way for Hausler to study the flexibility of the mammalian genomes is to run evolution in reverse, looking for what genes changed and when. Using a complex set of computational simulations, he's even managed to backfigure the genome of our common ancestor to an accuracy of 98%.
So the algorithm we call reverse evolution is a computer method for turning the clock back, as it were. The computer actually compares all 3 billion bases of the human DNA to all 3 billion bases of the mouse DNA, to all 3 billion bases of the dog DNA, etc. It's an enormous amount of computational effort. We have a very large cluster with more than 1,000 central processing units all churning away doing these comparisons. We scanned the genomes of animals looking for regions that hadn't changed for hundreds of millions of years and then suddenly changed a lot in the human genome. Suddenly, we found one. The most dramatically changed elements may be involved in the evolution of the human brain. Sometime in the last 3 million years, something changed in the DNA that caused the human brain to be much larger than its ancestral form.
This human-accelerated region 1 gene, nicknamed HAR1, now lies at the core of the team's ongoing research.
We take those elements of DNA and we take them into the wet lab and try it out in cells. You can actually change a whole gene and then look at how the mouse develops differently. We can even reconstruct the ancestral version of this gene as it existed 50 or 100 million years ago, and then see how the mouse behaves with the old version of the gene versus the new version of the gene.
In the search for the true nature of ancient animals, DNA is rapidly becoming the new dig site, and computers are the shovels.
My passion, what drives me to look at this, is to understand how humans became human, how a whale became a whale. The changes in the DNA will ultimately be the story that we understand of how the different animals that roam this planet today came to be the way they are.